19 Lab, Chapter 13. Most of this lab you can do at home on your own. The first part here is an excerpt from a book called The Man Who Mistook His Wife for a Hat. There's so much about the way your brain functions that you take for granted, and you can't really notice it because it's just always been there. But this is an interesting read of if you didn't have that processing center in your brain. So if you look at this page in the book, remember you have areas of the brain that receive the raw data. For example, if I told you to close your eyes and hold out your hand and I put an ice cube in your hand, the skin on your hand is going to feel that it's cold and wet and hard. That's the raw data. That information would be sent to your parietal lobe, to the primary somatosensory cortex. So it's going to get signals saying cold, wet, hard. Then your association area is going to interpret those signals. It's going to say, something that's cold and wet and hard, that sounds like an ice cube. It puts the clues together and it solves the mystery of what the object is. And you have that with all of them. So like, for example, with vision, you have the primary visual cortex. It tells you things like color, shape, size. And then your association area puts the clues together to figure out what it is. And back here in this story with the man who mistook his wife for a hat, he didn't have that visual association area. He could look at things and he could tell you what size it is, what shape it is, what color it is, but he couldn't tell you what it was just by looking at it. So the first part of the lab is about these areas. So you want to use page 498 in your book, and you can do this part at home on your own. So you're going to go through and you're going to do some little experiments. You're going to hold an object, just like I talked about, like with the ice cube. You're going to look at something. And whenever it talks about the raw data, that would be like in the ice cube example, cold, wet, hard. Which functional brain area receives this raw data? So with the one where we're doing your sense of touch, it would be the primary somatosensory cortex on the parietal lobe. What is the object? With my ice cube example, it would be ice cube. Which functional brain area and in which lobe use the raw data to figure it out? That would be the somatosensory association. Then you're going to look at an object. And this is going to be like the man in the Oliver Sacks book. You're going to describe the size and shape and color of it. That is going to be your primary visual cortex. Then you're going to say what the object is, like he was given a glove, and that is your visual association area that figures out what it is. Okay, then again, these next page of questions, you're going to use your notes and this page of your book. So having a thought, which lobe has thoughts? Speaking the thought, which part of the brain allows you to speak? Stand up and walk gracefully across the room. So made the decision to walk, so that's going to be from here. Which functional brain area and which lobe generated the nerve signals to send to your muscles? So think about what here is going to initially make those signals that go to your muscles and stimulate them to contract. Then which part of your brain coordinated the commands? With which part of the brain coordinated the commands, you have to think about other parts besides the cerebrum. So go back to those four main parts of the brain and think about the part that coordinates your activities. Then we have draw a work of art. So now you're using the part about the different hemispheres. Remember, you have the right and left hemispheres of your brain. And remember, it crosses over, goes to opposite sides. Again, question 6 is about the hemispheres, right and left hemispheres. Question 7 is back here to these areas labeled on this picture, and also use your notes and use this area down here too, is going to help you. Okay, here number 8, go back to the first page of notes, the main parts of the brain. Which part of the brain would have hunger, thirst? It has 7 jobs total. Hunger and thirst are 2 of them. Here, this is number 9, is about the hemispheres, the right and left sides. Number 10 is coming from here. Use pages 498 and 499. Number 11 is also from these pages. Number 12, the jump rope video, we'll watch that video now. And it's a video of some people doing a jump rope routine, and they have different color shirts, and your job is to count how many times the 2 women wearing green shirts jump. So here's the video.
All you have to do is keep track of the number of times that either of the girls in green jumps. You'll count each time one of them lands a jump, like this.
1, 2, 3, 4, 5.
As you can see, these jumpers are pretty quick on their feet, so you're gonna have to pay attention to keep up. When the whistle blows, start counting. Ready? Go. So, how many jumps did the green team make? Did you say 38? If so, you agreed with 40% of our test audience.
Okay, how many jumps did you count? Well, here's the trick. The number of jumps wasn't the real question. That was just a distraction to throw you off track. The real question is, did you see anything odd in the video? Did you notice anything? Some people did pick up on it, but most people have no idea. So we're going to watch the video again. This time, don't focus on anything. Just take in the whole big picture. So here's the video again.
All you have to do is keep track of the number of times that either of the girls in green jumps. You'll count each time one of them lands a jump, like this.
1, 2, 3, 4, 5.
As you can see, these jumpers are pretty quick on their feet, so you're gonna have to pay attention to keep up. When the whistle blows, start counting. Ready? Go. So, how many jumps did the green team make? Did you say 38? If so, you agreed with 40% of our test audience.
Did you see the chicken this time? It's pretty surprising. It's amazing how the first time that chicken was there, but yet you didn't see it. Now, there's nothing wrong with your eyes. Your eyes saw the chicken, and your eyes generated a signal about the chicken and sent that signal to your brain. But your thalamus is the filter. You have so much incoming sensory information that you can't pay attention to all of it. And so as all this information is coming into your brain, your thalamus is deciding what you need to pay attention to and what you should ignore. So because I had told you to focus on the 2 women in the green shirts, that was all your thalamus was paying attention to. It was ignoring all the other signals coming in. And that's why most of you did not see the chicken the first time watching the video. So your thalamus, and this goes back to in your notes, that page of notes where we talked about the parts of the brain, add this in on that page of notes. Your thalamus is the filter for incoming sensory information. You do not have the ability to pay attention to everything. You have to pick what you want to focus on, and that's what your thalamus does. And it ignores everything that you're not focused on. The next part is a little more information with the cerebellum. And you want to reference page 515 in your book, this clinical view box up here. It talks about how alcohol affects the cerebellum. And we're going to do some experiments with some drunk buster goggles. These are goggles that you can wear and it distorts your vision and it's meant to simulate being drunk. And alcohol inhibits the cerebellum. So these goggles are simulating inhibiting the cerebellum, and we'll see how these activities go. The next part is with cranial nerves. The point here is to figure out which cranial nerve is being used to do activity. So like, for example, smile, frown, raise your eyebrows, puff your cheeks. I don't want you to just put yes, that you're able to do those things. I know you're able to do those things. I want you to put which cranial nerve is making you do these things. So you're going to go through the next several questions and you're going to use your notes in your book to determine which cranial nerve does each of these activities. Some of the questions will have more than one cranial nerve listed, and some of the cranial nerves will be used in more than one question. Now we're ready to look at the brain model, then dissect a brain. So we'll begin with the brain model. So here is our model of the brain. And we're going to start with the main parts. So you have the cerebrum as the biggest part here. You have the cerebellum as this little part down here. Inside is the diencephalon and the brainstem. Then we have the lobes. So you have the frontal lobe, that is your thoughts, your personality, your decisions. Along the frontal lobe here, we have the primary motor cortex, which is stimulating your skeletal muscles. We have the parietal lobe, which is your sense of touch, and we have the somatosensory cortex that receives all those signals from your skin. You have the temporal lobe, which is hearing and smell, and you have the occipital lobe, which is vision. And the insula is down underneath. So remember in the picture in the book, it shows they have to pull back the frontal lobe to see the insula. And insula has your sense of taste and it's down underneath there. Okay, we also have Broca and Wernicke. Now this is kind of a trick question. This is the front, this is the back. This is the right side of the brain. And remember, Broca and Wernicke are on the left, so they're on the missing part over here that we don't have. But they would be right in this area and up here in this area, but on the left. And the corpus callosum is right here. And the corpus callosum is the part that connects the two hemispheres. The right and left hemispheres are connected to each other by this band of axons. That's how the right and left communicate. All right, here we have a sheep brain to dissect. So the first thing to notice is the meninges. So see, we have this covering here on the surface. This is the meninges that encloses and protects both the brain and the spine. We're going to go ahead and cut this, get it out of our way. Okay, and some other things to point out here before we start cutting. Notice these two little things on the front. Those are the olfactory nerves. That's what connects to the nose. And you can follow these back here to the temporal lobe. Then we also have underneath here some little stumps of what's left of the optic nerve. So this right here, and you can see they cross each other because remember right and left cross. So this is what's left of the optic nerve. It would have gone out like this and out like this, and then here you can see it's heading back to the occipital lobe. Okay, this is all cerebrum, And notice it has the gyri and sulci. So the raised areas are the gyri. The dense in between are the sulci. And the dura mater, remember, is the part of meninges that is the thin, delicate layer and adheres to the surface of the brain. So the dura mater is still here. That was the pia mater. The pia mater is the thin, delicate one that adheres to the surface of the brain. So here we still have the pia mater. You can see it. The dura mater is the one I cut away. So we have the cerebrum. Then here is the cerebellum, and here is the brainstem. Now this is from a sheep, which is a four-legged animal. So that's why the brainstem is pointing out this way. In a human, the brainstem would point down like this since we walk upright on two legs. We'll go ahead and we'll cut this open, and we're going to cut right down the middle here. And as you can see, the brain is pretty soft and squishy. Remember, it floats in that cerebral spinal fluid so that it's not crushed by its own weight. Now that we have it open, you can see the diencephalon is the inside part. This stripe right here is the corpus callosum. That's what connects the right and left hemispheres. And if we pull this open a little bit, you can see how that opens up and there's kind of a cave there. That is the lateral ventricle. That is the part that fills with cerebral spinal fluid. And if we open this up more, I'm going to go ahead and cut this like this. Okay, there's inside the lateral ventricle. Inside the lateral ventricle is the choroid plexus, the tissue that makes the cerebral spinal fluid. So you have, here's our other side just to review. Here is corpus callosum, and then this cave area back here that we can kind of open up and see is the lateral ventricle, and that tissue inside the lateral ventricle is the choroid plexus that's making the cerebral spinal fluid. So to build in a little quick quiz here, which part of the brain is this? Which part is this? What is this stripe of axons here that communicates between the right and the left? What is this open cave area here that would be filled with cerebral spinal fluid? Okay, then the last thing to look at here on our dissection is the gray and white matter. So if you look here, you can definitely see there's the white. That's the axons, and it's white because it's myelinated. Axons have myelin. Myelin is made of fat, and it gives it that white color. On the outside here is the gray part. Those are the cell bodies because cell bodies are not myelinated. And in the cerebellum, if you look inside it, the pattern of the white matter looks like a tree. And so that is called the arbor vitae. Arbor means tree. Think of an arbor tree. Arbor Day is when you plant a tree. And vitae means life. So this is the tree of life inside the cerebellum. All right, now we are outside on this beautiful April day, and Lance and Chloe are wearing the Drunk Buster goggles, and they are going to play catch with a football.
I'm going to throw it to the middle, to the middle one.
That's a dot.
Even just picking up the ball is difficult. All right. right, good game, guys. Why don't you give each other a high five?
Which one? There we go.
So the point of these goggles is that it's meant to inhibit your cerebellum from coordinating you.